Next topic is antibodies. What are antibodies? Antibodies, they are proteins that have a extreme high specificity for recognizing and binding to particular antigens. So antibodies, they are proteins that recognize and binds to the particular antigen. So antibodies, they are produced in by the immune response in response to exposure to the specific antigen. So when the immune system it encounters a new pathogen, it generates antibodies. So one virus or microbes can have several antigenic determinant sites that are known as epitopes to which the different antibodies may bind. What are immunoglobulins? Immunoglobulins, they are the glycoprotein that are produced by the immune system in response to exposure to the specific antigen. So they have ability to recognize and bind to antigen that triggered their production. So immunoglobulins, they are gamma globulins. They are also known as gamma globulins. Plasma cells, which are type of the white blood cells, are responsible for synthesis and secretion of the immunoglobulins. Immunoglobulins make about 25 to 30% of the total serum protein. So these can be found in various bodily proteins, tissues including serum, tissue fluids and the mucosal surface. Remember, all antibodies are immunoglobulins, but all immunoglobulins may not be antibodies. Now let us consider the structure of immunoglobulins. Immunoglobulins, they are composed of 4 polypeptide chains. Two identical light chains. So this one is the light chain, and 2 identical heavy chains. So they are linked by the disulfide bonds. So that are the disulfide bonds. They are the covalent bonds formed between sulfur in the cysteine residue. The light chains, these light chains, they have a molecular weight of around 25,000. So they are attached to the heavy chains with the help of, so this is the light chain and this one is the heavy chain, and they are attached with the help of disulfide bonds. So they comes in 2 variety. The light chains, they come in 2 variety. One is the kappa form and another is the lambda form. The heavy chains, they have a molecular weight of around 50,000. So the 2 heavy chains, they are together joined through dimer. And this association, it is mediated by 1 to 5 disulfide bond. Now each heavy chain has variable domain, that is VH, and 3 constant domains. That is CH1, CH2 and CH3. So this is CH1 region, CH2 and CH3. The light chain, it consists of single variable domain and single constant domain. So this one VL stands for variable for light chain and C stands for constant light chain, constant variable for the light chain. So remember Hing region. So this one is the Hing region that is situated between where CH1 and CH2. So it is situated between CH1 and CH2 region. So both the heavy and light chain, they have a variable region and a constant region. The variable region, it is responsible for binding to specific antigen. Whereas the constant, it is stable in structure and it determines the class of immunoglobulin. That could be IgG, IgM or IgA. So the variable region would determine the responsible for binding. Variable region is responsible for binding to specific antigen, and constant region, it determines the class of immunoglobulin. So within each immunoglobulin, the peptide chain, there are intra-chain disulfide bonds. So these there are intra-chain disulfide bonds that will form loops. So these loops, they are compactly folded to create globular structures that are known as domains. So the Hing region, we discussed earlier that it is region between CH1 and CH2, constant domain. So what is the function of Hing region? It will provide flexibility to the antibody structure, allowing it to move and adjust its orientation when binding to the antigen. So this is the light chain, and this one is the variable region for light chain. This is constant region for the light chain. And this one is the Hing axis that is situated between CH1 and CH2 region. Now the amino acid sequence of variable region of both light and heavy chains in immunoglobulins, they are not uniformly variable along the length. They consist of invariable region and highly variable zones. So what are the hypervariable regions? So within the variable regions, there are highly variable zones that are known as hypervariable regions or the hot spots. So these regions will play a role in formation of antigen binding sites where antibodies will react with specific epitopes on the antigen. Remember the hypervariable region, it contains complementary determining regions that are known as CDR. So the hypervariable region, it contains the complementary determining region. They are the specific parts of the hypervariable region that directly makes contact with the epitopes on the antigen. Each immunoglobulin peptide chain it contains internal disulfide links that are covalent bonds between the sulfur atoms in the cysteine residue. So these disulfide bonds contribute to overall structure and stability of antibody. Now papain it is a proteolytic enzyme that is derived from papaya latex. So it is commonly used to cleave the antibodies into smaller fragments. So when papain it is used on antibodies, it break the specific bonds in the hinge region uh, of the antibody. So the hinge region we know it's the flexible segment that connects the two arms of Y-shaped antibody molecules. So action of papain it results in formation of three main fragments: two identical FAB fragments, FAB stands for fragment antigen binding, and FC stands for fragment crystallization, crystallizable. The fragment antigen binding region it is part of antibody that contains the antigen binding sites. It consists of variable region of both the variable region of both the heavy and the light chain. So these variable regions they are responsible for recognizing and binding to the specific antigen. So FAB region it is crucial for specificity of immune response as it determines which antigen the antibody can bind to. The FC region it is the constant portion of the antibody molecule. So remember it is located in carboxy terminal of heavy chain. Unlike the FAB region, the FC does not this region does not possess antigen binding activity. Instead, it plays a role in biological function of antibodies. So this region it is involved in interaction with other immune components like immune cells and complementary proteins. Now the pepsin enzyme, we know it's a digestive enzyme that is found in the stomach. So when antibodies they are exposed to pepsin enzyme, it cleaves the antibody molecules in the hinge region just like the papain. However, the pepsin digestion it results in production of single fragment composed of two FAB units. So these fragments retains the antigen binding activity and they can bind to the antigen. Unlike papain digestion, in pepsin digestion the FC fragment it is typically not recovered intact. It is digested into the small peptides. That means that the FC region of antibody it is broken down into the smaller pieces and it is no longer available as the distinct fragment.